to our video on Lab 1B, diffusion and osmosis. This short video presentation will explore the concepts of diffusion and osmosis by reviewing the lab experiment. By reenacting the lab and reviewing the lab results, we hope that you will gain a better understanding of diffusion and osmosis.
For this lab, here are some key terms you'll need to know. Diffusion is the movement of molecules from an area of high concentration to an area of low concentration. Osmosis is the same movement, however, it is with water.
A hypertonic solution is a solution that has a higher concentration of solute than solvent. A hypotonic solution is a solution that has a higher concentration of solvent than solute. An isotonic solution is a solution that has the same concentration for both.
The purpose of the study of this lab is to find out which concentration of sucrose solution causes the most amount of water to osmose through the dialysis tubing. We hypothesize that the dialysis bag with 1.0 molar sucrose solution should have the largest percent mass increase, as it will cause the most water to osmose through the dialysis tubing. We have come to this hypothesis as the 1.0 molar solution is the most hypertonic and contains the least amount of water, which will cause the water molecules outside of it to rush in.
Here are the materials that we need for this lab. We have 6 beakers of 250 milliliters of water. In them, we have 6 different pre-soaked dialysis tubes.
We have our 6 beaters with our sucrose solutions, 0.2, 0.4, 0.6, 0.8, and 1, and our scale.
In setting up this experiment, we must tie a knot in one end of each dialysis tube.
And then in each dialysis bag, we will fill a different sucrose concentration in each bag, except for one which will be filled with water, and that is the control for our experiment.
Now we will proceed to add 15 milliliters of the correct solution to each dialysis bag.
The independent variable for our experiment is the concentration of sucrose.
The dependent variable is the percent change in mass of the dialysis bag.
Now the bag has been filled, you must remove most of the air with your fingers and then tie the bag, leaving a little room for expansion.
Now, after rinsing and drying each of these bags, we place them on the scale to get the initial mass and then record it.
After we have marked all the beakers with which molar solution is inside of there, we will carefully place all of them at the same time into their respective beakers. Water, 0.2, 0.4, 0.6, 0.8, and 1.0 molar solution. Now they sit for 30 minutes and we wait.
Has elapsed. We will remove the dialysis bags and dry them off before we can properly weigh them.
After they have been dried off, we will put them on the scale and measure their weight in grams and record it.
You find the mass difference by subtracting the first mass from the second mass.
You find the percent change in mass by taking the mass difference and dividing it by the initial mass, and then multiplying that by 100. In the bag with the distilled water in it, the first mass was 15.97, the second mass 16.45, the difference of 14.48 grams, and the percent change was 2.9%. In the 0.2 molar sucrose bag, 16.37 was the initial mass, 17.60 was the second mass, a difference of 1.23 grams, and a 7.0% change. In the bag with the 0.4 molar sucrose, we had an initial mass of 16.72, a second mass of 18.14, a difference of 1.42 grams, and a percent change of 7.8%.
In the bag with 0.6 molar sucrose, the initial mass was 17.36 grams. The second mass was 19.4 grams. The change was 2.04 grams, and percent change was 10.5. In the bag with 0.8 molar sucrose, the first mass was 17.69 grams. The second mass was 20.08 grams. The change was 2.39 grams, and the percent change was 11.9%. In the bag with 1 molar sucrose, the initial mass was 18.43 grams. The second mass was 21.2 grams. The change was 2.77 grams, and the percent change was 13.1%.
There is a very noticeable trend going on in the results of this lab. As the concentration of the sucrose increases, the mass difference also increases, as well as the percent change in mass.
Welcome to our discussion chamber. Through this experiment, we have observed that the percent change in mass is directly proportional to the increase of sucrose in the dialysis bag. This means that the bags with the largest amounts of sucrose had the most water osmosis to them.
For example, the dialysis bag with water in it had a percent change in mass of 2.9. However, the dialysis bag with 1 molar sucrose had a percent change in mass of 13.1%. Our hypothesis that the sucrose solution with the greatest concentration, which was the 1.0 molar sucrose solution, would cause the most osmosis to occur, was confirmed when our results showed that it had the greatest percent change in mass.
Now we'll discuss potential sources of error we could have encountered in our experiment. One potential source of error could have been inaccurate measurements of 15 milliliters into the dialysis tubing, which would have, as a result, given us inaccurate results.
Another source of error is that the dialysis bags could have leaked, causing the process of osmosis to be screwed up.
Lastly, we could have neglected to zero in the scales when taking all of our measurements, which would greatly disrupt our results. Another way we could have conducted this experiment would have been using a tube in which a similar membrane that water could pass through is used to separate the tube into 2 distinct chambers. One side would be filled just with water, and the other side would be filled with some sort of solution containing a certain molarity of sucrose. We would observe that the water would pass through the membrane into the other side of the tube, raising the level of liquid in solution on the other side of the tubing.
Based on the increased water level, we can determine which solution caused the most amount of water to osmose.
Diffusion and osmosis apply to biology in a number of ways. One of those is how cells use diffusion and osmosis to transport molecules that they need through their membranes.
Another way is how roots use osmosis to bring water into the plants.